All right, good evening. I'm going to tack on two mini lectures. Each of these is a topic and actually has to do with industry, so it makes a good mini lecture. And I want to thank you for asking questions today because I would much prefer to answer your questions and then we can have a little mini lecture rather than powering through when your questions remain unanswered. So I am very happy to do a mini lecture and do realize that a lot of times the mini lectures end up on the test as well. So the mini lecture that we have first is going to be about PKU. Remember that we had that one company that originally had a microbe that they would give to patients that would compensate for a broken urea cycle. That didn't work, but it turns out that their second idea, the one about a microbe to compensate for PKU, was working much better. It's not over yet, but it appears to be on the way. And so this is an idea that you have someone who has too much phenylalanine in their system because they don't have the enzyme that will hydroxylate it, that will be able to turn it into tyrosine, which is the only way to get rid of excess phenylalanine that you can't excrete, can't transform into another protein. There's one road out of it, and if that road is blocked, the patient has a problem. If they eat too much phenylalanine, it will accumulate and cause lots of problems. And so the idea is the extra phenylalanine naturally gets converted into other metabolites, which you can't really get rid of these either, but you can smell them on the urine. The idea is, okay, we've got too much phenylalanine. How do we compensate for this so that it doesn't end up as excess phenylalanine, phenylpyruvate, phenylacetate? And the company is Synlogic, and I can't even say the name, lavafenogene marcalocobac, but it's easier to call it SynB 1934, which is their microbe that they've actually designed and synthesized in a sense. And they put it into the small intestine, pretty easy place for a microbe to go and thrive. And what they do, this is a tweet list that comes from one of the scientists working on it, and this is often the best way to see what they're doing. It's treatment of PKU, which is going to be breaking down phenylalanine in the gut. And so they had actually originally tried SynB 1618, and they developed the strain SynB 1934 to improve on their first attempt at this. The product of the enzyme, so the enzyme that they have in the microbe that they have, is going to take phenylalanine and convert it to what they call TCA. I don't even have the full name of it, but you see that it's gotten rid of the amine, and now it has just a carbon backbone at that region. The problem is this particular step in phenylalanine degradation was accumulating because the product TCA would be inhibiting the activity of the enzyme. Very common, actually. If you have an enzyme that works really well, in a sense it can work too well because it can have product inhibition if the product itself is not taken down. So they decide to focus on that particular enzyme and to optimize it in the lab because, you can optimize the sequence of the enzyme and then put a better enzyme into the microbe so that it doesn't have product accumulation. What they did is they did an intracellular sensor for TCA because TCA was the problem. And so they were actually going to take their TCA and they did a screen. And once you have a screen like this, and usually they're based on some kind of green fluorescent protein, that's what they're showing here. And they put the green fluorescent protein gene that would be a sensor that would light up depending on how much TCA was in the cell. If it lit up, then they would know that there was too much TCA in the cell, but the cells that didn't light up would be the ones that they would look at more closely because the TCA was no longer accumulating. You see how you identify the molecule. Once you identify a molecule that is diagnostic of your problem, then it's pretty easy to have a binding protein that then interacts with green fluorescent protein, turns on that gene, lights up the cell. And the cool thing is you can do a whole library of cells. You can do evolution in a tube because you can put all these mutations into the organism that you're working with, and then the ones that don't light up, you can say, oh, I'll take that mutation, and then you can figure out what it does. It's literally evolution just accelerated in the lab. So they basically took the thing and it worked in the context of the cell that lit up, right? But of course, you don't know that it's going to work in the context of the human gut, whether it's going to work in the context of the purified components that you have. You need to do a lot of testing where you do different rounds. But the cool thing about this synthetic evolution type stuff is that you can actually do it again. You can get something that's a little better, and then you can do incremental increases. And you can see right here, they're comparing it to wild type, and round one is barely even above wild type. The fold over wild type TCA production is a factor of 1, but there's some error and there's some things that are better. So you take the ones that are better and you mutate them. You end up with things being climbing. Again, the average climbs, and you have some that are a lot worse even, but that's okay because you have some that are better. And so you keep doing, it looks like they did 4 rounds and they ended up with something that was 1.5 times better. You know, it's not 10 times better, but 1.5 times better was, they looked at that and they said that was enough for them. And so they did it in a different kind of assay, and again, fold over wild type for this assay, they did probably a different kind of mutation. You'd have to read the paper to really find out what's going on. But they did another angle, and they ended up with about 4-fold increase over wild type of the activity that they wanted, or in this case, the activity that they didn't want. I think it's the activity that either they're working on making TCA less, but they're probably working downstream on consuming TCA more. Again, I'm not doing this for sure, but then they took their improved enzyme. It's just, there's a lot you can do with just a sequence of an enzyme. They took this improved enzyme and they put it into their strain, and this is the thing that they show us is how the strain actually works. And you see you have phenylalanine coming in at one point, and you have a good uptake. A microbe that they've designed has a gene that takes in phenylalanine at a high rate, and then it has it converts it. It looks like it converts it to the TCA, and then the TCA actually gets ejected from the cell. It's possible that that is what they ended up evolving as well. They ended up some way to get the TCA out of the cell, so then you don't have product inhibition anymore if it's all leaving the cell. I'm just guessing from a Twitter thread, but you can tell a lot from a Twitter thread. The other thing about this is it's a couple of genes. They have, I don't know what the ARA-C gene is doing here, but I see that there's the phenylalanine P gene, then there's the MPAL gene, so that's 2 genes that they've put in. And it looks like they have some kind of surface protein, that's the 3rd gene that they've put in. But that's essentially what they have. They have 3 genes that they put into this microbe, and I don't even know what kind of, let's see, what species is it? Yeah, it doesn't say right here what the species is. Probably E. coli, but who knows, maybe they're doing that differently as well. The end of the story is, this is now going on to phase 3, and by the time you get to phase 3, you actually are starting to breathe easy. That it can actually possibly work, and this company can actually produce this product and help people and make money while you're at it. So that is story number 1, that it's going to be about this, to finish up chapter 18, whatever the chapter is.